Hi everyone, so in this video I'm going to go through oxidative phosphorylation and the electron transport chain, all of that stuff. So basically like when we're getting a good chunk of all of our energy. So I'm going to attempt to sort of like draw a rough idea of the electron transport chain just to give like a sense of what's happening. And so just to like lay out sort of where we are, because this is important, we're in the mitochondria, but we're right in the inner membrane. So this is the intermembrane space, sort of below this membrane. Here, I can draw the other. So this would be the outer membrane. This is the inner membrane. And this is the matrix of the mitochondria up here. Okay, so this is really where all of those reduced like energy carriers that we've made from glycolysis, from pyruvate processing, from the TCA cycle, this is where we're cashing that all in and we're getting a really huge ATP output. So you don't really need to know a lot of the details of what's happening here, but essentially you have like all of these sort of like complexes, protein complexes in the membrane, different like sizes, and then I'm gonna have to erase this. And then the important one obviously is the ATP synthase. Okay. Great. So overall, what's happening? So I'm going to draw in red everything that we sort of made like earlier. So for example, like the NADH that has like electrons on it now. So this complex will accept electrons from NADH basically through a redox reaction. So you overall get the oxidation of NADH and you're reducing this complex. So this complex is accepting electrons, it's reducing that complex, and then eventually like these electrons are sort of passed through the chain, and as we saw in class, we need somewhere to put these electrons on, right? They're passed through this chain, they're sort of like giving off their energy. They're coming from a really high potential energy molecule that was very reduced. And now as they flow through the membrane through these complexes, it's giving off this energy. And so at the end, these electrons will have to go somewhere, right? They're not just disappearing. And so as we saw in class, they're actually being put on oxygen to reduce that molecule to H2O. So O2 in this case is our terminal electron acceptor. And this is really important because when we talked about fermentation, the whole idea behind it is that we lose this terminal electron acceptor. And so none of this can work, right? Because we don't have anywhere for these electrons to go. So eventually all of these complexes in the membrane are just going to be fully like accepting electrons, and that's it. They won't have any like accepting power anymore because they're just like carrying electrons and they're sort of at capacity in terms of what they can accept because they're already completely reduced, and because there's nothing for them to donate their electrons to. So this terminal electron acceptor oxygen is very, very, very important. And so, okay, so now you might ask, well, where does all this energy go, right? We started with high energy electrons. They lose their energy as they travel across all these complexes, and at the end we put them on oxygen so that they have somewhere to go. So the really important thing here that sort of makes all of this work is as these electrons are, sorry, that's not actually accurate, as these electrons are sort of being pumped across, we're also moving a bunch of protons from the matrix into the intermembrane space. So as the electrons are losing their energy by going through all of these different complexes, that energy that it's giving off is actually put into the active transport of these protons. And so I know when we talked about active transport before, we were super clear on like, oh, it's transport that uses ATP. But in this case, it's sort of like active transport in the sense that these protons won't just diffuse on their own across the membrane. They're actually being carried across, and the energy that is being used to carry them across is the energy from these high energy electrons. So it's not in the form of ATP, but it's still an energy form, and the energy is from those electrons. And so as electrons are being pumped and pumped across, this allows this gradient to form. And before I go anywhere else, like more with this, I also just want to mention that we do also cache in our FADH2 molecule that we made in the TCA cycle. So this is also donating electrons. So overall, everything that was holding our electrons before, our NADH and our FADH2, are reducing our complexes, and those complexes will then sort of shuttle the electrons to the next complex over, sort of like down a line. And then as they're shuttled from one complex to the next, and they're losing energy at each stage, and that energy is being used to pump protons from the matrix into the intermembrane space. And eventually those electrons will find the terminal electron acceptor, which is where they'll become water basically, or turn like an oxygen molecule into water, and sort of they're happy like being there. And now we've established this really strong proton gradient, or H plus gradient. So this is really like the very key part of all of this, like why all of this works. And so, yeah, the main idea is that because we've established such a strong gradient, these electrons really, really, really want to flow back, or sorry, these protons really want to flow back across. So they want to flow from the intermembrane space back to the matrix. Obviously, like they can't, right? We've actively pumped them across. There's no easy path for them to go back. But if we gave them a channel or a path back, then they would like very quickly flow back. And that's really what ATP synthase is. ATP synthase is a path for them to flow back into the matrix. And since they're like so like the gradient is just so strong, they so badly want to go back, the flow of protons back through ATP synthase is what literally like turns ATP synthase, and that really is what's catalyzing this reaction where we're making ATP. And this energy transfer, like of using this electrochemical gradient to then generate ATP, that's called the proton motive force, and it's really just the idea that there's so much energy in this proton gradient, and that can be literally used as a force to move ATP synthase to get it to produce ATP. So that's sort of overall like what's happening here. But really the important thing to note is that you're just producing so much of an ATP output at this stage. So yeah, like from substrate level phosphorylation, remember we made 2 ATP molecules in glycolysis, and we made 2 ATP molecules from the citric acid cycle, so 1 for each acetyl-CoA. But at this stage in oxidative phosphorylation, when we cache in like all of the electron carriers that we've sort of built up to this point, we're making 28 ATP molecules per glucose molecule, which is a huge amount of ATP compared to the 4 ATP that we made up to this point. So really, this is so essential for the cell, because without this, like the cell is barely making any energy at all. So hopefully that illustrated sort of what's happening here. And the last thing I'll mention is just this is really similar to what you would see in anaerobic respiration. And so anaerobic respiration is occurring in organisms that are basically used to not having access to oxygen. So they've just been born that way. They know that you know, they just don't use oxygen for this process, and so they're doing something called anaerobic cellular respiration. And really, in this case, their terminal electron acceptor is just different. So instead of using oxygen, they would use, here, let me just write this down. So anaerobic respiration. So their terminal electron acceptor is some inorganic, oh my goodness, inorganic molecule. So like nitrates or sulfates. You don't really need to know these details, but you just need to know that there is a different terminal electron acceptor, that they basically have like a different way of getting those electrons from the complexes onto a terminal electron acceptor. So, and also generally, although they do something very similar, they have a similar cellular respiration like pathway, they're generally less efficient, and so they don't actually produce as much ATP as aerobic respiration does. But it's actually pretty cool how they have a very similar pathway, but at the end they really have a different terminal electron acceptor, which allows them to sort of do something similar. So hopefully this helped just give a general idea of what's going on. And in the next video, I'll talk about fermentation and how it's different from anaerobic respiration.